I think we'll start with the questions now, and again, encourage audience to send in questions. First question, I'll put it in for Sally, okay, which is, you've demonstrated now that looking at identifying cell types, cell function, cell phenotype, cell properties that could be that are critical for activity of our products, and identifying critical quality attributes. How would you try to implement a systems-based thinking? In what area would you think that a systems-based thinking could maybe have some low-hanging fruit? How would you go about implementing that in cell characterization? And, what kind of resources would you need? What would you needed to enable to do this?
So that's a great question, because clearly the sorts of data gathering that I spoke about for a cell product is not really compatible with a GMP process. I think it's more compatible with trying to really understand that product in depth in a more research environment. And we use these types of data analysis, high-level data analysis all the time when we take a product and we look at gene expression at the single cell level, for example, and what are the emergent properties that tell us about pathways and state of the cells. So I think that what we're also talking about, though, is a multimodal integration where you may have sets of omics data, like single-cell transcriptomics, but you also may have high-content imaging, live cell imaging, and computer-based imaging. And I think that one of the challenges is bringing together these different modalities so that ultimately we can make sense of it. In an earlier session, we talked a lot about emergent properties. Are there things that come from this type of large-scale analyses that are sort of fingerprints and help us to predict what will be there, what will effectively make this a useful and safe product going forward. So to me, then, I think that we really need to investigate the product thoroughly and probably do that in a research setting. Think about how to integrate the different data forms, and then ultimately, what do we pick for really putting through into a GMP process?
Yeah. And do you envision, like, through this process that you would be seeing, you'd be honing in on your critical quality attributes and even maybe revising them, tweaking them a bit as you're developing your product so that, you have a much better understanding of the function and the interaction of these particular cells in a human?
Yeah, and the challenge there, Jane, is that requires us to look at the patient and gather data from the patient and then bring that back and compare it with what we're finding out about the cell product. And so that's really demanding, from the patient perspective, from the cost of the clinical trial and study, what do we measure and for how long? But ultimately, we won't know what those critical quality attributes are until we bring the patient response and patient data in as well.
A great segue into, I'll turn it over to Doug now for a question on what do you think is the role of patients and patient advocates in activating or engaging or advocating for such systems-based thinking? And what would be the role of patients in actually participating in the data collection that could be used to input into such systems?
Thank you. Can you hear me okay?
Yes, beautiful.
All right, great. So just as a way of introducing my comments, I was patient number 2 on the initial University of Pennsylvania clinical trial on CART19. So my expertise is indeed as a patient. Back when I was treated, the only patients that had been treated were mice. So there wasn't a lot of knowledge, excuse me, on what to expect with humans. And one of the slides that was shown this morning showed the tremendous variability in response of patients to CAR T therapy, and not only response but toxicities. And I've tried to stay fairly close to what's been going on with CAR T over the years, and participating with LLS and with CMAT as a patient advocate. And I think the important part that a patient can play, obviously, is to be able to give feedback in clinical trials with regard to their experience. The effort that's ongoing now that I'm listening to in this workshop is extremely exciting from the standpoint of the efforts being made to try to understand and be able to predict what to expect with this type of therapy, because each patient is individual, but to be able to find the right parameters to measure, to be able to predict what the patient should expect, what the outcome will be, is really important. I talk to a lot of patients who are thinking about CAR T therapy now, or are about to start, and their biggest question is, what should I expect? What's going to happen? And the answer that they get from their physician, the answer that I right now can give is, every patient is individual. And there is a spectrum of responses to expect, but obviously I try to also give a message of hope. But the more that we can really understand what the patient's makeup is and how they're going to respond to it is very, very important.
Yeah. Switching to Adrian right now, you provided an outline of a beautiful system of data that you've already collected that can be used to, start integrating some of these thoughts, at least from the activity, functionality of your particular, of the CAR T cell therapies and different patient data that can be integrated. How have you moved this along right now, and what are some of the challenges that you are facing or have faced in implementing such systems-based approaches?
Yeah, I think that the most important challenge is the need to scale up and scale out the data sets in post-commercial setting. And we are trying to do that through harmonized biomarkers approaches and protocols, partner up with registries such as CIBMTR and other stakeholders, and make sure that we work with physicians as well as patients actually to have the appropriate consenting for additional data collection, as it is absolutely critical for us to identify, train, validate predictive algorithms for toxicities as well as efficacy in context of CAR T cell intervention. We have made a significant progress in that direction. We now know far more than 5 years ago in terms of the key determinants of response to CAR T cell intervention, but our job is not completed. We need actually to create very strong predictive algorithms based on systems biology thinking, and we cannot do that without partnership with appropriate stakeholders in this commercial post-marketing setting. So that's basically pretty much what we are doing. And we are poised to also partner up with academic collaborators, including UPenn, Moffitt Cancer Center, other major comprehensive cancer centers who are also collecting data. and deposit these data and analyze them in an harmonized fashion. Thank you.
Okay. Going to Bala now. Question, you talked about manufacturing systems. Again, similar question to what I asked Sally, where do you think systems-based approaches can be integrated into the manufacturing system, and how would you start doing that? Where have the challenges been in looking at such systems-based approaches from the point of view of manufacturing?
I want to kind of go back to, you asked the question to Sally about, what type of measurements can be made. And she talked about from the point of view of, that's more from the point of view of understanding and establishing the mechanism of actions that you're talking about. There is an intersection between design thinking and systems thinking. Design thinking is often involved in development, and in the process of development, one has to keep in mind how things are going to be manufactured. So while the measurement technologies that you do to understand things are more complex, one has to bear in mind that it is also possible to think about generation of biomarkers or surrogate measurements that actually provide insights into the mechanism of actions that you're talking about. So the way that I think about components of the system is not that you take what is being measured to understand a process at a fundamental level needs to be implemented on the manufacturing floor. For anything that needs to be measured in a manufacturing floor, you need to provide an answer in 15 minutes or so, because this is a continuous process. You cannot stop the process and come back 2 days later, or to say, well, the flow cytometrist is not available, for which I don't have multi-parametric information. And therefore, the measurements during a manufacturing process have to be providing information when and where required, so that you can take actionable information, actionable activities. So when you view it in that context, many, many tools are available today to be able to make measurements at each components of the process. Mm-hmm. And then based on that information, make adjustment in the subsequent process to compensate for it. It doesn't have to be an open loop system. It can be a closed loop system. And I think that the real point at which the interaction has to take place is right now, the information on the incoming material, there's very little characterization being done. Being able to move the process all the way to the cell procurement can make a huge impact in the overall control of the processes. So I feel that each of the systems, we can think in terms of making measurements. Let's take, for example, viral vectors. Right now, we look about simply in terms of the control on viral vector delivery, but we're not looking at receptor densities that are generated. How uniform? How does it vary from patient to patient? How does the impact on cell health has an impact on the survivability of the engineered cells, and so on? So over a period of time, these process characterizations will lead to a better understanding of the overall manufacturing process. And just as was covered in session 1, we need to understand the interaction, not just like the way we understand interaction between cells. We also need to understand the interaction between components of the system in the manufacturing as well.
Yeah, thank you. I think I'll open it up to the whole panel right now, a question that's coming in, which is, again, related to patients and patient variability. And that we know that, in particularly with certain starting materials and with a variety of different characteristics of patients, that can lead to a lot of variability. And the question is, are we collecting enough data in our clinical trials about subjects in those trials and about the particular aspects of our product to actually make good decisions or to make good assessments of integrating various aspects of either manufacturing or patient variability or et cetera into systems-based thinking. And what more data do we need to collect? I know, Adrian, you mentioned something about having to revise and even looking at getting more data. So I think the question is, how can we improve our data collection, particularly from patients, and how do we implement that?
I can take a first stab at that. I think it's a critical question, right? Because CAR T cells are based on patient's own immune system. And in the immune system is a collection of heterogeneous T cells and B cells and other immune cells. In this particular case, we're mostly interested in T cells. So the heterogeneity, patient-to-patient heterogeneity is overwhelming. And it is also a key key catalyst to understanding what are the most important attributes of the pre-treatment immune system that may influence product characteristics and thus clinical outcomes. In order for us to fully leverage this tremendous opportunity that we have, we need actually to go all the way to single cell omics analysis, right? In face of this heterogeneity, we need to understand, I'm afraid, at a single cell level, some of the key characteristics, such as the T cell fitness that I mentioned, the capability of T cells to expand, resist apoptosis or cell death, as well as engage multiple immune programs simultaneously, which seems to be also linked to clinical performance. And with all of this information, what we can do, we can build better predictive algorithms that are more actionable at the bedside, right? Because they're relying on pre-manufacturing characteristics of the immune system, as well as, equally importantly, pave the way to a new generation of cell interventions, which is based on designer cell, off-the-shelf cells, sourced from either stem cells that are differentiated ex vivo or from donors that are very well defined, thus reducing tremendously the interpatient variability in the next generation products. Thank you.
Doug, again, looking at, having collecting data from patients, do you see any challenges or things that could be better communicated to patients about, what we're trying, what various organizations, developers of various regenerative medicine therapies are trying to collect and actually getting an extensive panel of data from subjects who might be entering a trial? You're on mute, Doug.
How's that? So, again, I think one of the biggest challenges right now, again, from not only my own personal experience, but being involved with patients that are, as I said, contemplating this therapy, is trying to distill down all of the information that exists now, and there's a lot of it, and it doesn't, my sense of where the science is at the moment is that we have a lot of information, but we don't exactly know how to apply that to each individual patient to say this is the way it's going to go for you because you're XYZ. But we could do a better job in trying to distill down what we do know to provide that to patients sort of generally for patients that are thinking about going or going to go into CAR T therapy. I'm not sure if that answers your question, but it's it's amazing to see what's happened in 10 years. in terms of we knew absolutely nothing about what was going to happen, and now we've got thousands of data points. But I think, again, just continuing to collect each individual's experience and try to start matching that to biological parameters, and I think that's eventually we'll get there.
Yeah. So again, to the panel, do you think that collection of iPSCs from individual patients could be helpful as a tool in trying to tease out some of the heterogeneity that might go on from patient to patient?
So Jane, I think that is a really interesting idea now, because as we see pluripotent stem cell technology develop, our ability to produce many different tissue types is really enhanced, not just in 2 dimensions, but in 3 dimensions, to build organoids that are representative of gut and brain and skin and inner ear, and it's really quite extraordinary. And I think that those cells could be used to study the individual and to get at some of these questions. So I think it's a very interesting idea, and definitely worth thinking about. And no, I'm not sure in terms of the development of CAR T, how far we are in terms of making the relevant cells. One issue with iPS cells is that they do tend to be of an immature state. And that's certainly something that we would have to contend with. But that issue is being dealt with. We're learning how to mature the cells, and I think that they could be valuable tools in this process.
Great, terrific. Bala, did you have any comments?
Well, I was just going to make the comment that, historically we say that, we look for a lost key under the light, because that's where the light is. And oftentimes in cell therapy, what we're really looking at is we're making measurements enabled by existing instrumentation. We're not starting from the point of view, and this kind of a workshop is extremely valuable in that context, that what are the parameters that are important and that are going to define they are critical to quality, and what is the type of measurements that needs to be made. So my feeling is that I think that we're going to find development of new methods of generating information that is not in front of us. And it is that type of information, whether it is cytokine profiling, it's phenotyping, all of those things, and collecting that across not just multiple patients, but also across multiple types of drugs addressing the same disease state. Then we will be able to make sense out of how does the systems biology actually translates itself into impacting manufacturing. And that's the point I wanted to make.
Yeah, great. So again, I'll open it up to the whole panel, but in looking and thinking about these will create, such approaches will take tremendous amounts of resources. And do you think that a single sponsor, so for instance, Kite, for you, Adrian, can undertake such an endeavor, or is it something that, has to be across a consortium of different groups in order to be successful?
We started by launching a consortium with large academic organizations who participate not only to clinical trials, but also sponsor research, as well as post-marketing stage for both Axicell and Brexucell. We have 2 products approved to date. And that's the first step. making sure that we generate this data, this big data, utilizing a harmonized biomarkers protocol with schedule of assessments that are aligned, with methodologies and analytics that are aligned, with one single, let's say, structure, command structure in terms of data integration, as well as analysis, utilizing pre-specified protocols, as well as aided by machine learning. But that's not sufficient. We need to actually establish, in my opinion, a consortium across different sponsors, various sponsors and organizations in our industry. The other companies may be interested in our data, and vice versa. We need to team up with each other as well as with the regulators and make sure that we accelerate in real world the data generation so that we can pave the way to optimizing patients' management, both from the standpoint of increasing the rate of durable responses, as well as mitigating or predicting toxicities that may require intensive management in the hospital. Thank you.
So again, to the whole panel, getting back to the original title of this particular session, which is, what are the costs to the field and the field of regenerative medicine if we don't implement such systems? I'd like to hear from each of the panelists. Sal, you want to start?
Sure. Your question brings to mind a real-world example, which is from a company that was generating a cell therapy for spinal cord injury. And I think that we've begun understanding that changes in that manufacturing process or in some aspect of it actually led to a failure of that product. And while the product may have passed the criteria for release, it was clear that there were aspects of that product that were not being captured. And, cells are so complicated, as we've been discussing. But when you get into a clinical or GMP manufacturing process, everything is expensive and everything is pared down. And, it's been our experience, too, that while we would love to have a more comprehensive testing for release, it becomes prohibitive in the scheme of what you're trying to do. And so I think that the cost is really a cost of failure. It's failure of the enterprise, but most importantly, it's a failure of delivering something to patients that really could be beneficial. And so, I just can't underscore how I think we need to characterize these products much more carefully. And as we've been discussing, integrate that with patient data so we can predict responses better, but also identify subpopulations of patients that are more secure in their benefit.
Any other comments from the...
Yeah, I think if you actually look at the cost of not doing the systems thinking is going to be that these will be desperate, individualized processes. I mean, we have known that unless and otherwise we standardize these processes in some fashion so that you can indeed bring about a framework that people can establish competencies, etc., that you're not going to find that this is not a manufactured product, but rather an individualized patient-by-patient processes that will always remain unaffordable by a large segment of the population.
Yeah, I would like just to add, the price we will pay actually in the end is missing the opportunity to expand the footprint of potential curative interventions in early line and benefit actually as many patients as possible. So we need to continue to make efforts and embark on this road, systems biology thinking. Thank you.
Okay. So another question from the audience. Has anyone looked at ABO type and clinical outcomes with respect to CAR T-cell therapies? This is probably for you, Adrian.
I'm supposed to be the translational medicine representative, right? There are no significant associations between these type of parameters, including, let's say, phenotypic characteristics of immune blood cells and outcomes to CAR T-cell intervention outside what I already mentioned, the fitness of the T-cells, which are actually the backbone, the driving force behind the CAR T-cell intervention itself. So in addition to that, the dose of specialized T cells, especially those juvenile T cells in one's immune system, we call them naive-like, some people call them stem cell-like memory cells, relative to pretreatment tumor burden seems to be a major determinant of durable response after CAR T-cell intervention in B-cell malignancies, leukemia, as well as lymphoma. On top of that, and that's why we need systems biology approach, it is those fine characteristics of the tumor immune microenvironment that can be defined through gene expression analysis and gene sequencing, such as immune programs, interferon programs, chemokine programs, cytokine programs, and all of that, which actually influence the interaction between CAR T cells and tumor and determine whether a patient will achieve or not clinical response. So we have to go to a much higher level of detail and sophistication in terms of what we are looking at to define the predictive algorithms for CAR T-cell intervention, I'm afraid. Omics. Thank you.
Great, thank you. And we're over time already, so I'm going to conclude this session and thank all the panelists and the audience for participating. Thank you.
Thank you.
Thank you.